Right, hello everyone, hope you're doing well, and today we're just going to do a quick lecture on drug metabolism and let's talk about some of the medication that we use. So if you're in hospital, what you're given and how does the body metabolize those drugs and what it does with them basically. So we've got quite a lot to get through, so let's get straight into it. I hope you're all doing well. So if we kind of split up the action of drugs into four main parts. So we've got absorption, distribution, metabolism, and then excretion. So firstly, we talk about absorption. So that's basically the movement of a drug from site of administration into the blood, and that can be orally, intravenously, or other ways that we may not want to talk about. So we've got this gastric mucosa for oral drugs, rectal membrane suppositories, so those can be quite painful. Buccal cavity, so oral drugs again. Skin, transdermal patches, so something like a nicotine patch. And a pulmonary epithelium, so inhaled drugs, so such as asthma medication. Okay, so how do these drugs get into the body? So we've got these lipid-soluble non-ionized drugs, which are absorbed much faster. And we've got this effect of ionization. So basically what happens, so drugs tend to be either weak acids or alkali, therefore they freely get ionized. However, for example, we've got a weak acid drug, so therefore when there's high H+ concentration, wherever the drug is, that means it can't be ionized. And that means it can easily be passed into cells, and that makes them more lipid soluble, so they can enter something like enterocytes in the enteric system. What happens then is when they get inside, there's less H+ ions, for example, and it's mainly water, so they can therefore become ionized, and then they lose their solubility and they can't leave anymore. And the same happens with weak bases. If there's a lot of HCO3- and they are non-ionized, and then when they enter they become ionized and they're trapped there. So yeah, that all depends on this pH of solution, and therefore we get this ion trapping, which I just mentioned, where the drug enters, for example aspirin, in this non-ionized form, enters and becomes trapped when it metabolizes into this salicylic acid. And then this is in this lipid-soluble state, which is able to pass it through. And then it can be reabsorbed in the kidneys, but we'll get onto that later. Okay, so the other two, distribution and metabolism, we'll talk about those in a bit, but firstly just note on excretion and this increase and decrease in gastric emptying. What can we do to get these drugs out of the body faster or slower? And that could be either beneficial or not, depending if we want those drugs inside us to obviously help us out or not. So we've got this physical activity, alkaline for volumes, disease state, administration of large volumes of liquid, alkaline solutions, disease state, emotional stress, and other drugs can also interfere with each other. Then the decrease, we've got ingestion of food, physical inactivity, poorly soluble antacids, disease state, and emotional state, as well as other drugs as well. How do these then get delivered? So we talk about distribution now. So delivery of these drugs from systemic circulation to body compartments, mostly by blood plasma, but also bound to protein, such as this albumin that we will keep talking about, basically transports everything around. So some drugs are also quite special, which can cross the blood-brain barrier, so act on the brain or placenta if someone is pregnant. And what are the factors that basically affect where these drugs go? So we've got regional blood flow, so how much blood goes into certain parts. So obviously if we've got rich blood supply, the drug will work more in those areas. Again, lipid solubility and plasma protein binding as well. So how much affinity does the drug have to a protein carrier? And most drugs become inactive when they bound to this transport. So they basically get on this transport train of albumin, become inactive, so they can just chill out. And when they get to the active site where they need to work, they become activated again when they unbind. So we've got this human serum albumin. So we've got this binding of protein, which binds to acidic drugs mainly, and because albumin has an overall negative charge in the blood, but it can also bind basic drugs. But so basically what happens is it has high capacity for these basic drugs, so alkali, but has low affinity, so they don't bind very strongly. Whereas with acid, it's basically the opposite and has low capacity but high affinity. So there's no all saturated sites in the albumin. Then we've got this volume of distribution, which basically predicts how much drug will get into the body. So we've got this theoretical volume that would be needed to distribute a drug if it was found at the same concentration throughout the body as that of serum volume. So how do we work that out? So that's basically amount of drug absorbed, so in grams, and the serum concentration. Two different types of drugs, so we can kind of categorize them into. So we've got those with high VD, volume of distribution, will have mainly be non-polar, have high lipid solubility, low rate of ionization and low plasma binding, such as chloroquine. The low VD are basically the opposite, so polar, low lipid solubility, in its ionized state, and plasma bound, so such as heparin. So we need less of that drug to work, basically. So how are they then excreted from the body? We've got this maintenance of volume and composition of body fluids, and this controls through this acid-base balance, and also the elimination of end products of metabolism, so metabolite, also xenobiotics as well. And we've got this clearance of fluid cleared of drug from the body per unit of time. So how do these then drugs get out? So firstly, if we go to the kidneys, where everything is excreted and filtered, so we've got this glomerular filtration, which is proportional to the free drug concentration in plasma. So depending how much drug there is, depends how much gets out, obviously. Then we've got this proximal tubule, which is the active secretion of ionized drug into the lumen, allowing them to be excreted. But also we've got this tubular reabsorption, which is an active transport kind of process, so it requires ATP, and that basically allows for the drugs to be reabsorbed into the body, keep on and get excreted depending on the body's needs, and obviously the solubility of the drugs as well. So if we talk about this metabolism more, we can come back to it. So we've got this first pass metabolism, so the extent of metabolism occurring before a drug reaches the systemic circulation. So we've got this bioavailability, which basically kind of the F factor, so the fraction of drug dose that reaches the bloodstream in an active form. So when drugs enters the liver through the hepatic portal vein, it can either leave circulation or it can be excreted in the bile, and that basically depends on its molecular weight. So what's this affected by? So again, first this pass effect, metabolism in the gut. So any hepatic disease which can slow down or increase the action of the drug. And we've got these three metabolic phases, which we're going to talk about. So we've got these metabolic phases, which are initial modification, so kind of the changing of structure of the drug. And then we've got addition of larger group, and then transport processes. So drugs can either go through the stage 1 to 2 to 3, or they can do something like go through phase 2 first, which we'll see an example of, then phase 1, or they can simply just go through from phase 1 all the way out and get excreted. So phase 1, what happens? We've got this mainly done by the CYP450 enzymes. So an example of that is hydrogylation, where this hydroxy group is added on to change the structure. And what that basically does is prepares the drug for stage 2. And that's helped out by these mixed function oxidases, as it catalyzes the oxidation of lots of lipid-soluble drugs and steroids as well, and some other stuff. So we've got this main reaction here. We've got NADPH plus H plus ions plus oxygen with this lipophilic substrate, so the drug, and that basically makes us NADP plus, water, and metabolite, which is more lipophobic now. So what occurs in stage 2? So that's in the endoplasmic reticulum of hepatocytes, where there's easy access to this conjugation method, as well as the CYP450 enzyme, so everything's close together, so it can occur quickly. So the main method is we've got this UDP glucuronosyl-transferase reaction, where we've got this addition of glucuronic acid. However, this conjugation can also occur through methylation, acetylation, and sulfation as well. So there's different ways of the drugs to be gone through phase 2. And that basically works to make the molecules more polar and easier for excretion. So again, we talked about this exemption, so we've got isoniazid. So that basically happens when phase 2 takes place before phase 1, and that basically makes isonicotinic acid. And we also talked about this therapeutic window. So obviously we don't want to give someone too little of the drug that has no effect, but we also we don't want to give them too much, which has adverse effects. So just a quick note on this therapeutic window is basically we want to find that middle ground of how much drug we want to give to the patient. So if we talk about now inhibition, so when the drug goes through the whole process, works nicely, but obviously sometimes things go wrong. So we can either inhibit the enzyme which breaks down the drug, so the drug obviously has more effect, depending if it's a normal drug or a pro-drug, or it can induce it, in which the enzyme works twice as fast and eliminates the drugs twice as fast to get excreted. Or if it's a pro-drug, and what that basically means is when the drug first comes into the body, it's inactive, and it has to be gone through this process through enzymes to become active as a metabolite. However, normally what happens is drugs come in and they're active, they work on the body, then they get broken down by these enzymes for this phase 1 and 2, and then excreted. So there's two different ways here. So yeah, we've got this inhibition of CYP450 enzymes, and that's the main group of enzymes, so we give them the common name. And that results in if this inhibition in increased plasma drug levels, also prolongation of effective drugs, or toxic effects, especially if the therapeutic window is quite narrow. An example of that is grapefruit juice, which contains these furancomarins, which inhibits the CYP450. So in paracetamol, we've got this metabolism to NAPQI, which is toxic. But normally what happens is paracetamol is broken down by glutathione into a non-toxic molecule. But sometimes this NAPQI can build up, especially if you overdose on paracetamol. And what basically happens is all these glutathione saturates, and there's not enough of them to detoxify this NAPQI, and that can lead to hepatotoxicity. And to kind of check for that, after about 48 hours, you can check the AST and ALT, so your aspartate aminotransferase and alanine aminotransferase, as those are markers of liver damage. And we've got this potential increase of oxidative stress due to this glutathione depletion, causing mitochondria to maybe not be able to make ATP, and that can then cause necrosis. But the treatment's quite easy. You can literally give loads of this acetylcysteine to patients, as it doesn't seem to have toxic effects, so that can be easily treated. So then if we talk about the factors which affect our drug metabolism, so we've got these internal and external factors, as with basically anything you do in science. We've got this internal, so your genetics, age, so stuff like infants don't have developed CYP activity, and the same with elderly, which have a decreased activity of these enzymes, so the drugs aren't broken down as efficiently. Also gender, so we've got menstrual steroid metabolism, causes female to handle drugs differently, and as we get these more ADRs in females, and also your disease state, so if you've got liver failure, obviously drugs will have an overdone effect on you. External on the other side, so obviously diet, so what you eat matters a lot, and as we saw with the grapefruit juice, so the effects of depending amount of antioxidants, also plant toxins, so we've got naringin and quercetin, which are inhibitors of many enzymes, as well as barbecued meats, so induce the CYP1A1 and A2 activity, as well as alcohol, so as we know, this can induce and inhibit some of the enzymes, and that's basically due to your liver shutting down slowly due to cirrhosis. Also the environment, so we can have the effect on tobacco smoke, exhaust fumes, pollutants, solvents, and pesticides. So then just a little note on pharmacogenetics. So what happens is when you've got these enzymes, obviously things can go a little differently in you, so you can get different variations of these enzymes which either work not well enough or too much. So we've got these base changes in gene chromosomal locus, and that may result in the changes in the production of receptor proteins, enzymes, and transport proteins, and that can affect all parts of those ADME that we talked about, so your absorption, distribution, metabolism, and excretion. So if we talk about some of the examples of what happens, so we've got isoniazid, so that's a prodrug, so as we talked about, so that's inactive when you take it in, then it gets muddled up by your enzymes, goes through those phases and becomes active, and that's used to treat tuberculosis, and it's activated by this bacterial catalase peroxidase found in them. Then we've got this, this acetylation of polymorphisms. So what happens is hereditary is very important in isoniazid elimination when you take in. And we've got difference between slow and fast acetylates. Is the drug basically broken too fast or too slow? Similarly, we've got DEBRISOQUINE, and that's a hypertensive drug. We've got the CYP2D6 enzymes. We have the CYP2D6 enzymes involved in this pathway, and metabolism of a large number of prescribed drugs. And again, we've got this variability in enzyme is cardiogenics seen in fraternal studies, fraternal twins. And basically what happens is this guy can either have none or duplication in his enzyme. What happens here? So we've got this 4 types that you can have of these polymorphisms in the enzyme, and that basically works for any enzyme that we talked about. So we've got poor metabolizers, where you're deficit in both alleles, so we've got no activity of the enzyme. We've got this intermediate, which you're defective of one allele, so the enzyme's still kind of working, there's some expression of the protein, but not much. Then we've got extensive, which is just your normal. And you can also have this gene duplication, where you have kind of double the enzyme, and then the drugs are basically removed from your body before they can even have an effect. However, again, in the case of this prodrug, if they're broken down too quickly, we get a head load of the active form of the drug metabolite, and obviously that's what happened to this guy, where he took too much and it had a mad effect on him. And then we talk about morphine. So codeine is kind of less potent than morphine, and this kind of the pathway. So about 10 to 15 goes to this norcodeine, 50 to 70 goes to this main codeine 6 glucuronide, and morphine is about 5 to 15%. So we know that most of the drug that we give people as codeine, only a little bit will be turned into this morphine to relieve pain. And again, we can get a mutation in the CYP2D6 enzyme, as we talked about, and which can again either increase metabolism of the drug and either have adverse or good effects on us. And again, it's a highly polymorphic gene as well. Then last we've got tamoxifen, so that's used for endocrine therapy, and it's seen that breast cancer may be due to variability in the CYP2D6 enzyme and a role in formation of INDUCTION, which is a harmful molecule, and it's seen to be interpreted in this breast cancer formation. So we've kind of gone through a lot today. I tried to summarize as best I could because there was loads that you can obviously go in depth on all of these, but I tried to give a quick summary of what happens in this drug metabolism. So I hope you enjoyed. Just have a look through, take your time. If you want to research anything and go into detail, please do, to understand the subject better. But yeah, thank you for watching. Leave a like, subscribe, leave a comment, all that good stuff, and yeah, I'll see you in the next one. Cheerio.